Genetic variant discovered in Amish protects from heart disease. Agene variant initially uncovered in the genomes of people belonging to the Old Order Amish has been linked in a new study to lower levels of fibrinogen, a blood clotting factor, and low-density lipoprotein (LDL) cholesterol, both of which, when elevated, increase a person's risk of developing cardiovascular disease. The work, published today, December 2, in Science, not only connects a missense mutation in the enzyme coding gene beta 1,4-galactosyl-transferase 1b4-gal1 with heart health in humans, but confirms the link in mice. Due to the small pool of genetic starting material, this population harbors less genetic diversity than the general population, meaning that variants that might disappear in larger groups can be maintained in Old Order Amish populations and therefore be easier to spot. In the new study, Montasser and colleagues sequenced the exons of 6,890 Old Order Amish subjects and found a missense mutation in B4-gal1, which is expressed throughout the body and encodes an enzyme that transfers galactose to proteins. The variant was associated with lower LDL cholesterol and was present in 6% of the Amish genomes, but is much rarer outside the Amish community. The researchers found it in only 8 of 140,000 non-Amish genomes that are part of a national heart, lung, and blood institute database. When the team looked at other factors related to cardiovascular health in people carrying the missense mutation, they found no association with triglycerides and a small association with high-density lipoprotein cholesterol, the blood clotting factor fibrinogen, which can be a risk factor for cardiovascular disease when elevated, was lower in people with the variant. To assess whether the missense mutation in B4-gal was linked with overall cardiovascular health, the researchers shifted their focus outside the relatively healthy Amish population to the Jasinger Health System and the UK Biobank, two larger genomic databases. Because the specific variant they identified in the Amish is so rare in the general population, Montasser and her colleagues pulled out genomes with any variant of B4-gal. These individuals had decreased LDL, fibrinogen, and a 3 to 5% reduction in cardiovascular disease. Before getting to the point of clinical relevance, we have to make sure that there are not any harmful side effects associated with having this variant, Montasser explains. Having low LDL is great. Having low fibrinogen is great. But is there anything else harmful? So far, based on all the other information we have, everything looks good. Those people look perfectly healthy, but we are doing even more deep phenotyping right now to make sure that we are not missing anything. Montasser says they will keep working on it, and hopefully someday we'll have some form of therapy based on this.